Okay. Here we are, four of us, five of us actually.
Okay.
Yeah, four in here with me.
Hi, I'm Patrick.
Patrick. And
Okay, so could you please explain just your background so that it will be easy to explain something? So, hi, Patrick, if you explain a little bit about your background, like what did you do before, like did it project or any thesis work or any, what is your experience in machine learning things? Just in a, like shortly.
Yeah. So I did a maths degree, majoring in statistics and operations research, which finished a year and a half ago, and then went straight from that into the Master of IT, majoring in data science.
Okay.
So I learned a bit about machine learning for the first time last semester in the core units as part of the degree, and then I made a recommender system in Python for my project last semester. Experience of some of the bits of the tools we're using, but on the whole is all new. Like I know nothing about medical research or.
Okay, that's fine. So do you have any experience about using R scripting language?
Yes, did lots of R in the statistics major.
Good, that's fine.
Started Python last semester in 3/4 of my subjects were Python based.
Okay, that's good. Okay, thanks. Okay, next.
he used to work with Yuzu, just down to like at some point, and Yuzu used to be here is the data scientist. Okay, so associate professor Yuzu.
Yeah. Oh.
Yeah. So I talked to her the other day. And, well, so I think he's very fit with the project. Okay.
So actually, I forget about the details. So I don't know so if he is experienced in using any programming language and a little bit confident about some programming, any programming language, even it could be Python or because Python are almost similar type of scripting language, and it is very easy compared to the any object-oriented or C++ or Java language. So maybe he can learn easily within a short time if he is interested. And since our plan is to develop some machine learning model and use those machine learning models on the biological data, you don't need to know in details about the biological things. I will give the required information to you, like for this project, because time is limited. Yeah? So we will do like developing some model or applying some model directly on the data. I will explain the data format and data details so that you don't need to read the biological book or biological literature. Yeah? You have to read when you will like when you will finish the project, or you have to write something, or you have to write the thesis or project details, project report, then you have to read some materials, but you don't need to learn in details about the biological things. So if he thinks that he's confident, that's fine. So it's not very hard. Yeah.
Okay, sure. And next is Ben.
Oh, hello. I don't know if the mic can pick on my voice very well, but I'm quite loud, so it probably does. I'm Ben, and my background is that I have a bachelor in pharmacy. So
biology.
Pharmacy.
Pharmacy. Okay.
Yes.
Yeah, so I'm a pharmacist, and I did some pharmacy work over the years, and over time I started speaking with software engineers or people related to e-health or bioinformatics, and I've noticed that it's sort of a field that I really want to try get into because I've always loved technology and pharmacy, but nowadays with more computational medicine, as we call it, coming out to the world, I can finally see it being realized where two fields of profession are kind of melding together as one intertwined profession. And so a lot of my knowledge is in the biomedical side for this side of project. When it comes to technical aspects, I'm much more knowledgeable in C#. So with Python, I have like the basic understanding to kind of string the syntaxes together to get this pseudocode running. But I wouldn't say I'm experienced in Python, unfortunately.
Okay, no. Do you have any experience about R?
I've heard about R. I haven't used it personally, but I've seen in the listing that R may be required, so I looked into how I can try to use it. So
Okay.
I did tinker around with it, but I can't say I'm good at it.
Okay, so like in this like the project that we are planning to continue, I think for completing this project, we need to learn some scripting language like R. Even we can apply other programming language, but R is very important for this type of project and bioinformatics area. Even for machine learning, we can apply Python, but I think that R, if you would like to pick some existing code, then R will be suitable for us. Yeah. So R is not very hard. So if you can learn it in a day or in a week, but like for initial purposes, you can start with the initial knowledge in R. Yeah. With the basic knowledge, you can do better. Yeah. Okay, that's good. So thanks.
Thank you.
Hi, I'm Lin. My background is my major is Master of Information Technology, and my bachelor is also the same. And I did some data processing and data management before, and last semester I used Python to complete my project, and also I tried some projects on the GitHub. So, but I don't really know much about the R. So for some research, I did some like entry level research before, but not experience like this unit.
Okay. Okay. So if I think that according to your description, like last semester you have learned some Python and applied some Python on some data analysis, but R is easier than Python.
Oh.
Yeah. So you can like easily learn those things within a short time, but it is important to learn R. Yeah. So okay, that's good. No problem. Maybe we will take time for learning those R scripting language, but it's fine. Okay, thanks. Hello.
Hi, Doctor. How are you?
Yeah, good. How are you?
Good. This is Anadeep. I'm studying masters in data science here. I have actually, I studied biology in my +2, so I got a bit of knowledge about biology as well. And in my previous semesters, as a part of artificial intelligence, I use R as well.
Very good.
So little bit I know about Python as well. And, I worked as a software developer for almost 3 years in India.
Fine.
So I'm into my final semester now.
Okay. So now, actually, my name is Muhammad Ali Moni, but you can call me Moni. So I work in Sydney University. I'm a UCIT fellow, like DVC fellow of this university and conjoint lecturer of engineering faculty. So actually, my background is computer science and engineering. Then I did PhD in bioinformatics and machine learning, as well as data science. Actually, my project included the clinical bioinformatics things. So for you, I thought that the single cell sequencing data would be suitable for your project because we can apply different machine learning models on those data. So to analyze, and we can develop some even we can develop some tools, or we can develop some new model, or we can integrate some model for analyzing those data. So this is the thing. Now maybe I need to show you the basic idea of the data. What will be the structure of the data? Then that will be clear for you what we can do. So I'm sharing my screen. Can you see my screen?
Yeah.
So actually, we will work on the biological data. That is the first point. So according to our project description, you saw that our project is based on the cancer data. It could be other data, but it would be the biological data related to any disease. Are you clear? So the data that I will provide to you based on the any disease or any biological data, it could be either human data, human sample, or mice sample. So either human sample or mice sample. So the similarity between human and mice genetics is about 70% is similar between human and mice. And this 70% is very important for any disease progression and also for the development of health and everything of the human. And other 30% which is not similar between the mice and human, that is the phenotype things like the shape, size, those things. So that is not important to learn or to understand for you. Just you need to know 70% is similar between human and mice. That is the key point. Yeah. So for this reason, we can exchange or we can analyze both data together or separately. Even in the last part, we can compare between the mice and human. Because why do we need to compare between the mice and human for implementation some drug or for application of some drug to any vivo model? We need to apply it to the mice. We cannot apply it to the human directly. Yeah. Because then if someone dies, then who will take the responsibility? So we need to apply to the mice. So for this reason, mice is important model for developing drug, for analyzing some data, for doing any work. So for initial study, we consider the mice, and for final study, like the clinical trial, we need to apply some drug in a small cohort of human. That is the last part, when we already know the outcome of the drug or something, then we apply it to the human. But in case of identify the positive reason, we can take sample from the human. Most of the cases, we can take all the part of the human body, but except the sometimes brain and sometimes some organs. That is the point. Now, what is the structure of the data? Actually, we will work. You can see that in human body, you know, there are DNA. Yeah? Do you have idea about the DNA? DNA. So actually, DNA, after the transcription, it becomes RNA. Is it complex for you? Because maybe most of you know the DNA, because DNA is very long, like a strip in human body, which has double helix, and it is like 3 billion. It is combined with 3 billion bits, and if we make some combination or permutation, then it will be 2 to the power like 3 billion. It is very big. So we are not going to explain those things. Yeah, just the DNA after the transcription, it becomes RNA. And after just 3 things we need to learn, then after the translation, it becomes protein. So this is the 3 point: DNA to RNA, RNA to protein. What is the DNA? In DNA, we know there are 4 letters: A, U, C, G, or A, U, C, T. These 4 letters makes DNA. Like if you, it is this DNA sequence, if we do the sequence of any DNA, then it is combined with, like it is presented with the 4 letters. Yeah. So that is DNA. So, but DNA is very long, like a strip. And if we take any part of the like DNA from any tissue or any cell of the human body, it will be same. Like it is same for even if you consider the hair or if you consider the tissue from hand or leg, it is same. Like all are similar, not similar, same. The sequence will be same from the tissue that we can consider from everywhere of the body. Yeah. So the DNA sequence is same, but RNA is not same. If we take RNA from the hand, and if we take hand means the tissue from the hand, if we consider the tissue from brain, then it will be different. So it is tissue specific. RNA is tissue specific. Similarly, protein is also tissue specific, but DNA is not tissue specific. So what is the difference between the DNA, RNA, protein? So DNA, from the DNA, we know that after the transcription it becomes RNA, but DNA is combined of two things. One is the active part, another one is the inactive part. Active and inactive. So it is very, so if you see that, it is very long strip, yeah? Very long strip DNA. And some part are active, and like if we see this one, so this is the DNA, yeah. So you can see this one.
Yeah.
This is the DNA, but is this DNA? So there are some active region. This is like the DNA, and it's like a strip, but it is a double helix, like two strip combinedly makes DNA. So then there are some active part, like this is active part, maybe this is inactive part, this is active part, this is inactive part. So when it becomes RNA, so we can see clearly some are active part and some are inactive part. And the active part actually works for everyday's work, like everything. Most of the cases, it works for everything. If you would like to raise the hand, then the active part works for raising our hand. How it works? Then the active part of RNA becomes, like after the translation, it becomes protein. When we think about to raising hand, then it like there is transcription, like the RNA, when it becomes the protein, and then it works to raise the hand.
Yes.
So because I'm trying to explain within a short biological idea, like a layman, so for this I'm explaining in that way.
Mhm.
So now we will work on only the RNA, not the DNA. But we will also work like only the very small part of our work will be related to protein, but most of the work we will do based on the RNA. But I told you that the RNA, so it is like when it becomes RNA from the DNA, there are two parts, like not two parts, like several parts. One section is active, another section is inactive, another section is active, another section is like it is a combined of active and inactive section. So the active section, almost we have 30,000 active section, almost, not the fixed. Sometimes it is more than 30,000. Sometimes it is less than 30,000. It is important point. What do you need to remember? There are about 30,000 active regions. 30,000. So the 30,000 active region of RNA, we call it gene. Gene, Gene. So these 30,000 genes have their own names. So 30,000 genes have 30,000 names. For example, BRCA1, maybe you heard this name because Angelina Jolie, maybe she suffered the problem of the BRCA1 gene mutation,
Yeah.
the breast cancer, yeah.
Yeah.
it is inherited from her maybe mom.
Yeah.
So this is the about 30,000 gene. So we will work only these 30,000 gene. Now, if you forget the the explanation that I explained before, if you forget those things, don't worry. You don't need to remember those things. Just we are now in gene. So I told you the basic idea because basic explanation about the DNA to RNA because you need to know something not very deeply from the how it comes from DNA to RNA. But RNA now we are talking about the active region. We are not talking about the inactive region. Inactive region, maybe most of the cases, more than 80% is related to inactive region, but active region is about 20% or less than that. But all it is about 30,000 genes. But you need to remember there are 30,000, almost 30,000. It could be 25,000 or 35,000. It depends on the different technologies. So just you need to remember near to or about to 30,000 coding region. So we will work on these 30,000 gene. Now, if we take sample from a cancer patient, like breast cancer patient, and if we like 5 breast cancer patient, and if we consider 5 healthy patient sample, like even consider the blood sample. Then if we compare these two sample, two group of sample, like 5 breast cancer, 5 healthy women. So if we compare these two samples, then we will see that there are some gene are dysregulated, like differentially expressed in cancer patient compared to the healthy people. Maybe it could be 20, 100, 200, I don't know how many. So we can say those 200 genes which are dysregulated for this breast cancer patient are the biomarker of the cancer, breast cancer. Is it clear for you? Because it is the first day I'm explaining these things. Then you maybe you can ask some questions related to this one.
If you don't have any questions, please.
Because it is I'm explaining taking some small part from different section and trying to give you the required information, not the in details, the proper lecturing things. So for this it is sometimes difficult for me also. So so these 200 genes are the biomarker. Sometimes we call it signature genes. So now I have a question. So if we give always you have to think about the numerical value for each gene. So if I would like to give you 5 sample healthy people data and 5 sample from the breast cancer patient data, and the feature will be about 30,000. Because 30,000 gene information. So what will be the matrix size? It is basic question. What will be the matrix size? What will be the data size?
Any idea?
Yeah, it is easy because 30,000 is your feature, 30,000 genes, and 10 sample, 30,000 by 10. Yeah.
Yeah. Yeah, they know it. They know it.
30,000 by 10 because there are 30,000. We are considering what is the 30,000 gene information. So we are taking 30,000 gene information from the 5 healthy healthy people data and 5 cancer patient data. So we will have if we put in a CSV file, then we will get 30,000 row and 10 columns. Yeah. So is it clear for you?
Yeah, they're saying okay.
It's like maybe I can show you one example data that will be clear for you. Oh. So I'm opening a data set so that it could be
more easy to understand.
to understand for you.
That'd be good.
So can you see a CSV file? Yeah.
Yeah.
So like for example, if we remove, okay, so you can see this one, or I need to make it bigger. So you can see this one, yeah.
Yeah.
So you see that this, like this is the identifier or gene symbol, yeah. This is the name of the gene. So there are about 30,000 of these gene symbol, and these are the like the sample from each patient. This is one sample, this is another sample, this is another sample, this is another sample. So this is the sample, yeah. So there are many samples here. So this is the gene, like for example CD3Z. It is a gene symbol or gene name. Yeah? It is a gene symbol or gene name. We can say identifier, but it is gene symbol. So the difference between the gene symbol of mice gene and gene symbol of human gene is the letter, like the representation is different. Like for human, all letters should be capital, and for mice, all letter like only the first letter is capital and others are small letter. So this is the mice sample. Yeah, for this reason, the first letter is capital and other letters are small character. Okay, so this is one sample. This is another sample. This is another sample. This is another sample. This is another sample. So there, if I give you this type of spreadsheet or CSV file with the 5 sample from the healthy people and 5 sample from the breast cancer people, can you identify or can you do some statistics based on this data, like which feature means which gene is significantly difference between these two groups? Is it possible for you? Yes, you have to apply some t-test or some statistics. Yeah, then you can find out which gene is showing significantly difference between these two groups. Then you can say that, okay, this group, maybe these 10 genes or 100 genes or 200 genes are significantly different between these two groups. That means those 200 genes may be related to the breast cancer progression or related to the breast cancer. Yeah, is it clear? So our first identification will be like that one, like to identify the significant genes. That is the first point of bioinformatics, to identify the significant genes or significant biomarkers. So it is very easy. For example, I am showing another data sets. Can you see this second data set?
Yeah.
Yeah?
Yeah.
Yeah.
So in this data, like after analyzing the previous data, not the exact the previous, but like similar data, then we got this result. Like we tried to compare two groups for cancer and non-cancer group. Then we tried to find out which genes is related to the cancer. So just we have applied some t-test and we have tried to find out the adjusted p-value of each gene for two groups, like p-value, adjusted p-value, t-test value, Bayesian p-value, and the logarithmic fold sense. So based on these statistics, we try to find out which genes are significantly related to the breast cancer. Is it clear?
Yep.
So it is very easy. We have programming code. Just we need to run those code in R, especially in R, because all code in R is freely available. And for statistics, it is easy to apply or easy to implement in R. So that is the point. We can try to identify the significant genes. Now our point is, like, we are working only the coding region of RNA, and the coding region is about 30,000, near to 30,000 coding region, and 30,000 coding region have their own names. That is called the gene symbol. So you need to remember these things. And so if we give any data to you, there will be about 30,000 rows. Yeah, 30,000 row, and the column will depends on the number of sample. Is it clear?
Yes.
So if I give you this CSV file or any spreadsheet file, then it is easy to apply any method on that data. So this is the thing. This is the background. Now we are going to our real data explanation, because we are planning to apply or develop some method on single cell sequencing data. So before I told you that the RNA data is different, like it is different cell to cell, it is tissue to tissue. Like if we consider from the sample from the hand or sample from the blood, and if we consider sample from the brain, then it will be different from each sample to another sample, right? Even from the same patient. Not all gene will be different, but some gene may be different from one region, one organ to another organ. And we know that in one organ, like each organ has some combined of a number of tissue, and for each tissue, it has more than one cell, or sometimes it has one cell. It depends on that one. So till now, the drug companies, they develop drug based on the bulk sequencing data, bulk sequencing information that I showed you. Bulk sequencing means I have considered the blood sample from the breast cancer patient and blood sample from the healthy people. Then we have tried to identify the significant genes, and based on the significant genes, then the drug company try to develop the drug. That is called the bulk sequencing. We do not think, okay, from where, like how the gene works on the cell level, because if we take the blood sample, then it is combined of different types of cells. Yeah, so in blood maybe there are 30-40 types of cells. If we take the maybe sample from the breast or ovarian, ovary or from the brain, then maybe it has maybe 30-40 types of cells, but we don't know how many cells in each type of tissue. Or like tissue means if we take anything from the hand, that is the tissue, but it is combined of number of cells, but we don't know how many cells exist in on tissue. That is the problem in our study. We will try to solve this problem in our project. We don't know how many cells in our tissue, but we need to identify, we need to cluster based on the data to identify the number of cells. So that is the need. So we need the computational things like clustering algorithm to identify the number of cells. Is it clear? So we need to develop something, or we need to apply something to cluster to identify the number of cells from the tissue. So now I'm going to explain the single cell sequencing data. That will be our real data. Till now I explained the general idea of the data, and the general actually it is the bulk sequencing. The data comes from the sequencing. I'm not going to explain the sequencing thing. Just when we consider the blood sample or any sample from the human or mice or any other models, then we need to send to the. There are many biological process. Even I don't know all the process. There are many. You need to make. You need to use robot or any pipetting or anything. Then you need to send to the sequencing machine, and they do the sequencing. After sequencing, we need to apply different method, different tools to come, like to make the data like a CSV file. Yeah, but I will provide to you the CSV file, so you don't need to think about the
Data.
like previous process, the complex processes. Yeah. So we will provide the CSV file. So the first part, you don't need to do anything on the first part. Yeah. So we are going to do the bioinformatics part, but it is called the the functional analysis or statistical analysis, this part. So we will provide the CSV file. So now the single cell sequencing thing. So before I told you that if we consider the blood sample from the breast cancer and the non healthy people, then we can compare the gene between these two groups. Now our question is, if we would like to know the cell type, like we have the blood sample, then there is a technique to separate the blood sample into cells. Like we can separate the blood sample, we can make cell by cell. We can separate the blood sample, any sample, cell by cell. There are many methods to make it, like 10x chrome or TrueSeq, DeepSeq, NanoSeq, many methods to separate the sample into cell. So if we, like before, we considered 5 samples from the healthy and 5 samples from the patient. But now if we separate the cell, separate into cell, like if we consider one sample from the healthy or patient, then we need to separate into cells. So there is a technique, there are many techniques. We can separate just after taking the blood, then we need to we need to make the DNA, and we will not do, but the biologists, they actually do that one. Then after making the DNA, then we need to convert into the RNA, and from the RNA we will pick only the coding region. The coding region, number of coding region is 30,000 almost. So we got it. Now that is the point. But nowadays people are thinking in different way. They would like to make the drug based on the cell specific, because you know that in cancer patient, if we provide the drug, but after 5-6 years they usually die, or sometimes they live longer, but it has huge side effects. For this reason, people are thinking to consider the cell specific drug, so that it is useful for the patient and it will be helpful to reduce the side effect. So in this case, after taking the blood sample, then what you have to do? You have to separate the blood sample. It is a tissue, blood sample, into cells. But you don't know how many cells you will get. maybe after making the cells, you get maybe 5,000 from on sample. Yeah, so 5,000 cells. So you have considered 10 sample, but now you are getting 5,000 cells from each samples. How many sample you will have then?
30,000 times.
Yeah, 5,000 multiplied by 10, because you are dividing the tissue into cell. So, and from the each tissue you are getting about 5,000 cells. Yeah. So now your column will be 5,000 into 10. Yeah. Because you are making on sample from the cell specific sample you are making. It is little bit tricky. Okay, I'm explaining again. Like when you consider the blood sample, for example, on sample, then on sample, before doing anything, the biologists, they try to separate the cell. They make on cell, separate cell by cell. Yeah, cell by cell. So they, like on drop of blood, they try to, from the on drop of blood, they try to separate cell by cell. So maybe you can get 5,000 cells, sometimes you can get 4,000 cells. It is not fixed, but not types of cells. I'm talking about the total number of cells you may get from the on drop of blood. Using TrueSeq, NanoSeq, DeepSeq, 10x Chrome, many methods. So you are getting the cell specific data from the tissue on drop of blood. It could be anything, on drop of blood, even sample from the ovary, sample from the breast, sample from the brain, anything, sample from the anywhere. So you can separate into cells. So when you have separated, you don't know which the name of those cells. You don't know. Then the biologist, after processing, many pre-processing, like from extracting DNA, RNA, then they need to send to the machine, sequencing machine. After sequencing, you get cell-specific information, like I showed you for example, okay, I showed you this one, yeah. So this will be like, for example, so this will be on cell, another from, like I'm considering on sample, yeah. Now on sample, only talking about on sample, on drop of blood, and from on drop of blood you got maybe 5,000. Maybe initially I can tell you maybe 10 cells. You got 10 cells. So when you sequence, you do the sequence, and after finishing the pre-processing part, then you will get for 10 cell information like 10 column. Yeah, because it is cell specific information related to each gene, not the tissue. Before you got the one tissue, and you got for on sample you got the 30,000 gene information. Now you are getting cell specific because you have separated the cell. You have now the like 5,000 cells. So you are from on tissue, on sample, you made 5,000 sample. On sample to 5,000 sample because it is separated. But you will, before you got 30,000 gene for on sample, but now you have 5,000 sample because 5,000 cell. But maybe for all sample, for this 5 sample, you will not get 30,000 gene information. Maybe you will get 2,000 information. You get for some gene, for some cell, you will get 20 gene information. But now you will have 5,000 column, yeah, for on sample. So here you will get the 5,000 sample column. So from on sample. So if you have, if you do not understand, please ask me, no problem. So if you have 10 samples, then you will get maybe 10 into 5,000. We cannot say 10 into 5,000. Maybe 5,000 plus 4,000 plus maybe 3,500. But you are not sure how many cells you will
So
get
gene,
from on tissue.
yeah, so gene varies, gene will vary. So variable is
It will
gene.
vary. That
Yeah.
is the point, because how for that is the reason you can do the clustering.
Yeah.
So now from 10 sample, you will have maybe 5,000 into 10, 50,000. So your data will be 20,000 by 50,000. Yeah. So it is now big data. Maybe some cell, like in that some cell will be 0. For example, here you got 0 because you did not get information. So because now you are doing a small part, like cell is a part of the tissue, like it is divided into 5,000. So now the question is what you will do. So now you have to do the clustering to identify, to group the cell. Like there are you have now 50,000 cell. Yeah, from 10 sample you got 50,000 cell, but you need to cluster those sample into cell specific. So if you now you need to identify the cell type. So before identifying cell type, you need to cluster. So but you don't know how many cells you found in your tissue. So which method is suitable for this? I have a question. You don't know how many tissues, how many cells. How many cells means how many types of cells. Yeah, you need to now identify the types of cells. So now which type of machine learning model, like clustering method, is suitable for this? Supervised, semi-supervised, or unsupervised, or which one?
Unsupervised. Someone says unsupervised.
Unsupervised, but we don't know. But people are not successful till now about the application of unsupervised clustering on this data, to this data. So people are now applying some semi-supervised. They are trying to make, okay, we may consider 30 or we may consider 25, like that one. Yeah, they're trying to tune after clustering and changing the cluster number of cluster, which is a problem. Yeah, which is a problem of this clustering. Yeah? So you need to solve this problem. Who can do that one? So you have to work on this. You have to run the existing methods, existing codes, and trying to identify their problem in that code. Like they have just applied different type of unsupervised or semi-supervised or supervised method. They did not develop new method. You know, you do not need to develop new method. Maybe you can apply deep learning. You can apply the any convolution neural network or any type of things and try to identify what is the problem. How can you solve this clustering problem? Okay, that is one question for your research. You need to understand.
Yep.
The one question, you have to solve this clustering problem. Okay. And to solve this problem, how can you solve this problem? Step 1, you have to run all the existing pipeline. It is freely available, freely accessible. All are, most of them, 99% are written in R. I already shared with you Surat, which is the best now. Till now, people are thinking this is the best. So I shared with you that algorithm, that like the scripting language. Yeah? All code, all tutorial, everything is given there. Yeah? So at first you need to run this on. Similarly, you have to run, there are other maybe 10 to 20, 10 to 20. And you have to see that which method they have applied and what is their problem. You can compare those things and you have to try to find out the problem, and you can apply new statistics. You don't need to develop new method. Maybe you can apply the existing new approach, like maybe graphical convolution neural network. This is the new approach. You can apply that on. Or you can apply maybe deep learning. People are thinking that on. You can apply any other convolution neural network or artificial neural network or anything. That is the first question. So to solve this problem, you have to run all the existing code and try to understand that, and you need to think which one is suitable or is there any better approach to apply that on. And when you will run, you will do those things. You have to keep all the records and the text. You have to write something because you have to submit some report. Yeah. So like at the end, if you need to write the report, then you will be in trouble. So when you will run this thing, what you found out, and which program you have run, everything you have to write in a document. Okay. So this is the first question. And how there is another option for people consider this on. That will be your second question. Like people consider only top 500 gene for each sample. Like each cell, there are maybe, you don't know how many gene information you will get, but they consider for each cell top 500. So top 500 or top 700, it is not fixed. People don't know how many they should consider. So they consider 500 or 700, but you need to think how many. It should not be fixed. Like computer science, statistics, data scientists, they cannot make it fixed without any evidence. You need to think how many genes we should consider for this clustering. This is the second question. It should be dynamic. Maybe you can consider this one based on the variance or based on the distribution, based on the binomial distribution, negative binomial distribution, or uniform distribution, variance, mean. Like you can think all these. So how many genes you have to consider? Is it clear?
Yep. Yes.
Okay. This is second question. Third question. Third question. All of these are statistics and machine learning related. Yeah? You don't need to know any biological things so much. So third question is, okay, you have finished the clustering. Yeah. So each cluster, like for example, you have 1,000 cells, 1,000 cells, and you have clustered maybe 50. You found that okay, 50 cluster is fine. So on an average, in each cluster contains how many cells? How many cells? 50 cluster, 1,000 cells.
Could you repeat the question, please?
On an average, how many cells exist on an average in each cluster? That is your. I am asking you.
20 on average.
On an average.
Yeah.
20. Yeah, 50 into 20 equal to 1,000.
Yeah.
So 20. So each cluster you are getting almost like 20 or 50. You don't know, but on an average 20, according to this question. But now you have to. This is another question. Like this is the third question you have to solve in your project. So I'm giving a specific question. Yeah? Maybe you can get additional question. You can maybe you need to try to solve additional questions. Yeah.
Yeah.
Third question is, okay, in each cluster you found, okay, maybe in one cluster you found 20 cells. Yeah. And these 20 cells, how did you consider? based on this data set, you are considering based on the expression of each gene of that cell. Yeah. So now you need to calculate the power. So this third question is power calculation. So power calculation, what is the power calculation? For each cell, you have to identify either the probability or distribution of each cluster and try to find out what is the confidence level or power of that cell to exist in that cluster.
Okay.
Today, this is the 3 questions I am providing to you to solve these 3 questions from your data. Now, the method, you see that you need to know 2 things, only the statistics and machine learning methods. That means the statistics. Yeah? So clustering approach and some maybe distribution and some test like the probability test. You need to know the variance and distributions, probability. Even you can apply some Bayesian approach. Any clustering approach, you can apply the deep learning, supervised, semi-supervised, unsupervised, reinforcement, or any type of machine learning things you have to apply. This is the first part of the work. And next, I will explain the second part in another day. If I explain today, you will forget it. So I have provided 3 questions to you.
Okay.
And you have to solve these 3 questions. But initially, you have to run the existing programming code. And the people who do not know the R scripting, you have to learn something, the basic scripting in R.
Yeah.
It is not complex.
Shouldn't be.
And I will provide all the link of the code. You have to run those code, and it has also manual like the tutorial things. So you can solve it easily. And when you run this code, maybe you can share each other. You can learn from each other.
Yeah.
And that is the point. So initially, I have shared 3 links. One is like the basic presentation from MIT about the data, and you can go through that lectures. And then I have shared another link for the presentations on the single cell sequencing. And third link also from the second, like the same authors, about the code and the tutorial and the manual, everything is there. So you can run that things. So maybe you can run those things in a week. I will, because you have to run about 20, yeah. The first one will be harder for you because others are similar, all are similar. Some people applied like Pearson correlation, another person used maybe another type of correlation or Euclidean distance measurement, like these things, yeah. But for clustering, maybe some people applied PCA, some people applied T-SNE plot, some people applied maybe other type of statistics. It's not very like new things. So, and you can solve this problem in online code. If you write the Pearson correlation, it will give you in R the Pearson correlation. It is not hard. Yeah, online can solve your statistics. You don't need to write the proper programming in R. You just need to put the maybe the proper functions. All are built in. You can use some packages, R packages, bioconductor packages. So it is easy. And all the tutorial, all the materials are there. After finishing this one, then I will give you proper data for you. Because the sample data is included in this tutorial, so you can solve that one. Maybe from the MIT link, you cannot download the link data. That is, you don't need to worry about it. maybe for Sirat, you can do that one. So that's fine. Huh?
Yeah.
Cool.
That's
Is it
good. Yeah. So the data set basically they will follow from the tutorial, right?
Yes, initially from the tutorial,
Okay.
and then like when you will be confident, then I will give you the specific data for each person, not the common data for all of you. Because on data sample, it is about $5 million.
Yeah.
Yeah. If you can make very good approach, or you can solve something, or you can improve the existing method, then I will give you our own real data. That is $5 million for sample, this sample. It is very expensive data, yeah. And if you think that okay, you are just analyzing those data, then maybe I have to give the either publicly available data or the data that I have already worked. Then I will give those data. Yeah. So yeah, but I will do the first part. Like I will give you the maybe CSV file so that it will be easier for you to solve within a short time.
Okay, that's great.
Otherwise, it will take time. Yeah, for a PhD, it will be a PhD project.
Yeah. That's the point. That I was
Maybe
talking.
after finishing that, and you can do the PhD on that. No
Yeah.
problem. And it is easy to get a scholarship for PhD in this area, and funding is available in
Yeah.
single.
Sure.
Okay. Do you have any question?
Any questions?
No.
Feel free.
Okay.
Now they are going to digest these things. And
Yeah. So if you have any question, you can send email. No problem.
definitely.
Yeah.
Any questions?
Also, yeah, go through these three, and your timeline, like you have to finish in a week.
Yeah.
Because it is not hard.
We're expecting you to utilize.
Even I will learn from you.
Yeah, so research is something like that, you know.
Yeah, it's not like I know everything, you don't know anything.
My
You know
professor
better than me.
He's a professor here and one of the like senior professor, but well, so he's exploring some area with me, and sometimes like after criticizing me, he said that I learned a lot from you. But that's after criticizing. Anyway, so yeah, there's the thing. So the point is, we do have presentation the next week, right? Which that's a very important thing. I want all of my students should have get the highest grade. I try to help them, but the promise is like that. If you maintain our requirement, then well, eventually you'll go to your desired things, and that is the win-win game. You learn, and something will happen. Good things will be outcome. So. Have you registered for week 3? Already registered. Okay. So I think the storyline you have get from here, right? So you have to tell the story there, and you have to present based on the story. So the moderator can understand that what actually you are going to do. So don't put there like only the technical things, but tell the storyline, like what the single say, and be short of the story. Okay. Yeah, and meanwhile, we really expect you to spend time with these projects, 24 hours. If you are able to do something good, any stage we do have different level of publications. In any stage publication, it would help you to get a good like prospects, both in academia and industry. All right?
Okay, yeah, that's fine.
The point. So. Yeah, so reach out anytime regarding your problem. If you have any questions, mail to us, both of us. And yeah, that's it for today.
Great.
Yeah. So when will be the next meeting? So maybe you can take your decision.
Yeah.
So yeah.
We'll talk and we'll let you know. Sure?
Okay, thanks. Okay, thank you.
Thank you so much for your time.
Okay, thank you.
Thank you.
Okay, thank you very much.
Yeah.
Okay, bye.
Bye.
Bye.